So
Is it recording?
it's recording. So we're
Awesome.
gonna start with number one. So
Should we introduce ourselves?
yeah, let's do that. I'm Owen.
Okay, I'm Catherine, and we work together as a group for these problems.
Yeah.
So I guess what we're gonna do is just go one by one answering the questions, and hopefully we got them right.
Yeah, letting you know a little bit of how we did it. So on question one, let's see here. There you go. On question one, it said, suppose you perform the following cross. Gives that to you. Assume that all these genes undergo independent assortment. And so what we did was, instead of doing a Punnett square, it was the fork line method. I think that's what it was. It had a fork in it, but went through and figured out the probabilities for each allele, right? Each gene, and then we figured out the probability. So at least with the A's, you'd have a 1/4 chance of having a homozygous dominant, a 1/2 chance of getting, or 50% chance of getting a heterozygous, and then a 1/4 again of having homozygous recessive. And so we went through and listed those probabilities, and then at least for A, the first one, there was a 50% chance of having this genotype, a 1/4, or 25% of having this genotype, a 1/4, 25%. And we went through, and then you multiply across all of the different possibilities, and we got 1/128 was the proportion of offspring that would have that specific genotype. Then instead of going through all of it again, the B, C, and D, we got 1/256. C is 0, and D is 1/32.
Yep. So for question number 2, it was fill in the blanks, and that's pretty self-explanatory. We got a lot of the answers from the textbook as well as the PowerPoint. But for A, it says paired homologous chromosomes are sometimes called bifins or tetrads. For B, is the structure that connects the centromere to the microtubules of the meiotic spindle is called a kinetochore. For C, a diploid fruit fly has 4 pairs of chromosomes. A fruit fly cell in prophase 1 has 8 sister chromatids, while the fruit fly cell in metaphase 2 has 4 sister chromatids. For D, a test cross is a type of genetic cross that can determine if a purple flower plant has the PP or the homozygous dominant P or the heterozygous genotype. For E, a non-functional allele is sometimes called a null allele, while a partially functional allele is called a silent allele. And F, Mendel's law of independent assortment involves 2 traits, while the law of segregation involves a single trait.
Cool. And so I guess it is my turn for
question 3.
For question 3. So the question was, suppose an organism has the genotype homozygous dominant T, heterozygous R, and heterozygous Y, and assume that all of the alleles assort independently. Asked to draw a diagram of the homologous chromosomes arrangements in the organism during metaphase 1, and make sure to note the alleles. And so this, let's see if it'll share. This is the picture I ended up drawing. And so I just listed the possible alleles, possible genotypes across the top and the bottom. And then since it's in metaphase, they line up on that metaphase plate. And then I guess the different ways it can align is this one I kept the same, added a recessive Y and just kept doing that until I ran out of, I guess, alleles that I could change. possible gamete genotypes that can be produced from this organism would be down here. So I went through and wrote all these down and I guess took out the ones that were duplicated. So you could have all dominant genes, partially dominant with a recessive Y, a dominant T with recessive R and Y, and then a dominant T, a dominant Y, and a recessive R. And so during metaphase 1, there are 6 sister chromatids found per cell. And then during metaphase 2, there are 6 chromosomes in there as well. Since, yeah. Number 4, yes.
Let's see. For number 4, it says a horse has 32 pairs of chromosomes. And then for part 8, how many chromosomes are present per horse cell at the conclusion of meiosis 1? So with this one, we got 32 because at the conclusion of meiosis 1, first it has gone through, it already has split up, and then the DNA already replicated itself. So then at the conclusion of meiosis 1, it split up, and per horse cell is equal to 32 chromosomes. For question part B, how many tetrads are present per horse cell during prophase 1 of meiosis? Again, that's 32. For C, how many sister chromatids are present in a horse cell during prophase 1 of meiosis? We got 64. For D, how many sister chromatids are present per horse cell during metaphase 2 of meiosis? We got 32 as well, again. And for E, how many bivalents are present per horse cell during anaphase 2 of meiosis? We have 0 because at the end of, during my anaphase 2 of meiosis, there's no longer any more bivalents forming. So those were our numbers that we came up with, chromosomes.
And so for this number 5, it said consider the following tetrahybrid. Let me add the question. So to give us the genotype that we're crossing and gave us what each allele, I guess, for what each gene was. And they asked what proportion of the offspring will be tall, green, and terminal. And so what I did is I ended up listing all the different, see if I can do this. I ended up listing all of the different, I guess, possibilities of each genotype happening. And then when they gave the what the phenotypes would be, we knew that you would have, at least in this case, for A, you would have, let me see here. You would have 2 possibilities because if a tall could be homozygous dominant or heterozygous, and so you had to create 2 genotypes that would happen and create all those different phenotypes. And so we end up getting 9 out of 64 for A, 1 out of 32 for B, 1/16 for C, and for D we did 1/64. So I guess with that one, what you have to watch out for is if a dominant phenotype is there. If it's showing something like that, then you have to, I guess, make note that showing off that dominant trait, it can be homozygous or heterozygous. So, yeah.
And we pretty much just distribute it between the 2 the two genes, the crosses that they gave us. Again, using the forklift method.
Yes.
And there was our little presentation of our group work.
Hey. All right. Bye.
Can you send me the link so